Thanks everyone for showing up and attending. Really excited today to welcome Dr. StephanieHicks, who's going to talk to us about scalable statistical methods for software for single cell spatial data science. So a little bit about Stephanie. She is an associate professor in the biostatistics department at Johns Hopkins University. And a faculty member of the Johns Hopkins Data Science Lab. Stephanie completed her undergraduate degree. I have all this here from LSU, and then did her master's degree and her PhD at Rice University. She was a postdoctoral research associate in the Rafa Lab at the Harvard T.H. Chan. And got a K99 RO Pathway to Independence Award. Stephanie has done some really groundbreaking work in biostatistics and has really this holistic focus on both interesting statistical methods and building tools that are actually usable. I don't remember exactly what you follow your website, but sort of problem-driven research, problem-forward research. And so, you know, in addition to all these really interesting statistical methods, Stephanie has really taken the lead on developing resources for the community that have been incredibly influential, like the Orchestrating Single Cell Analysis open textbook in R, which many of you may be familiar with. Stephanie is recently the recipient of the award from the American Statistical Association for Teaching in Health Sciences Young Investigator Award, as well as the COPS Emerging Leader Award from the American Statistical Association. So we're very excited to have Stephanie here today to tell us about this recent work. And without further ado, please take it away.
I just try to get this connected in like show mode. Okay. Yeah, I
think I remember.
Oh yeah, you can hide. I learned this in my class this fall. Right, I forgot. Sorry guys. You can do I think the state. We're gonna get there.
Hey.
Oh, the magic button, right? Okay, thank you. All right. First of all, thank you for the invitation to join you here today. I am very excited to talk about my research. So as noted, I'm an associate professor in the Department of Biostatistics, but my research really spans outside of biostatistics, classic biostatistics. So I'm heavily involved in projects where I find there are interesting biological questions. And in particular, I'm really interested in genomics. So my area of expertise has sort of been specialized now for quite some time in single cell transcriptomics, but within the last year or two, my lab is sort of moving into methods and software for the analysis of data. If you've heard about that, I will give you a little brief introduction later if you haven't. So that's kind of where I'm at. Does this work like this? Okay. So if you are familiar with gene expression data, the classic way that most people would encounter this, at least when I grew up in the world of my graduate work, was bulk RNA sequencing. So the idea of bulk RNA sequencing is you take a tissue, say from somebody's post-mortem human brain. It has millions of cells in it. Those are heterogeneous cells. And what we get is a molecular readout for one observation for one bulk gene expression profile, which is an average expression across all the cells in that tissue for each gene. But again, like those tissues are pretty heterogeneous given by like the colors of the different cells in this tissue. So the way I like to think about it, I have young kids, I have a lot of Legos in my house, is imagine it like a big pile of Legos. Where maybe the width of each Lego is a particular feature and you just get to see an average of widths across all the Legos. Fast forward to 2014, 2015, when single cell technologies came online. This was a huge advancement in the field for gene expression, where now the readout for every observation is a single cell. So we get a molecular readout of, let's say, the full human transcriptome, so all 20,000 protein coding genes for each individual cell. And so you get to kind of see the different cellular resolution, maybe the colors represent different cell types. So the analogy is, it's like taking our big pile of Legos and being able to separate now into nice little piles. Maybe you get information about the different types of cells that are in your population. You get information about maybe the relative proportion of each cell type and so forth. So more formally, beyond Legos, single cell RNA sequencing is the process where we take a tissue and we isolate and disassociate individual cells. I'm not going to go into like how one does that. There are a lot of technical ways of doing that, but we can sequence each individual cell. And for simplicity, I'm going to take the liberty of fast forwarding it. So at the end of the sequencing experiment, you get a readout of what I'm going to call reads. And we just have to figure out where those reads map to, let's say, for example, in some kind of reference transcriptome. If I did my math right, there are 18 barcodes, there are 18 reads there. And so that number 18 gets recorded in a matrix. So we get a read, an observation or an observation for 18 reads from that map to gene 1 and cell 1. Maybe this one has an enormous expression, for example. And then a lot of this matrix is actually 0. So in single cell RNA-seq, this matrix is between 50% and 99% 0, where in bulk RNA-seq it's much, much less than that. And then this matrix can do a lot of cool things. So for example, we could plot gene expression and see which genes are differentially expressed maybe between cell types. We could apply dimensionality reduction techniques and see how they are different in some kind of low dimensional space, maybe a linear space, such as principal components analysis, and so forth. All right, that's single cell. Very quickly. Who's heard of the Human Cell Atlas? Some? Okay. So the Human Cell Atlas is this really amazing worldwide organization where made up of like thousands of labs, it has to be, where the goal is to create a map, essentially a molecular map of the human body. And they want to be able to understand all the different types of cells in the human body. So you can imagine single cell RNA-seq is a perfect tool to be able to do that. So you could, for example, create a map of the human liver across different donors, the map of the human brain, and so forth. In 2018, they released a draft, like a white paper, where they said as an initial draft, we want this map to have 10 billion observations or cells. So just like imagine putting that into a matrix, 10 billion observations. Let's say we're interested in genes of the human, so like 20,000 features. That's a big matrix, right? It's a really big matrix. That's a pretty extreme example. You may say, okay, but there's not like multiple human cell atlases. How much is like an average data set these days contained, or like how many cells are we sequencing in an average data set? So I'm showing you 4 data sets. This is from 2019 and 2020, I believe. All right, 2019 and 2020. And in red, I'm highlighting the number of cells or the number of observations that we're sequencing in these data sets. So on average, 2 million plus and so forth. This information was cherry-picked from this really cool website by Valentin Sensen and Leora Pachter, where they created like a manually curated database of all single cell publications. Last I touched it was in 2022 or like early 2022. So I don't know like how up to date it is for like today, but it's a really great resource if you're interested in like finding single cell data sets. So hopefully this like convinces you that like we're having larger and larger data set, or like we as computational biologists and statisticians and computer scientists, for example, are dealing with larger and larger data sets. So one of the challenges that my group is focused on is to develop algorithms that scale up to really large data sets, for example. And so I'm going to take a dive into unsupervised clustering, specifically with k-means, but then I will move on and talk about other areas of my research. Okay, so when I started in this area, when I started working in this area, we didn't standardly have 2 million cells in a particular data set. We had, a couple thousand, but it was very clear to me that like we were moving to a space where millions of cells are going to become an issue. So I started working with some collaborators, Elizabeth Burdham and Debbie DeRousseau, to sort of build or to answer the question, if we wanted to build scalable clustering methods for millions of cells, for example, what do we need? So the first answer was that we needed to have data formats that we could work with on disk or in memory. This is new for the statistics field. Typically, we're very used to working with data in memory. And so having like on disk formats being brought to this field was like really exciting. The file formats that you might be familiar with are HDF5, ZAR, LDE. These file formats are widely adopted and used by the Human Cell Atlas. So if you go to the Human Cell Atlas portal and download data, these are the formats you will encounter. And then, of course, we want it to be fast. Like we want clustering algorithms to be able to work with data in memory or on disk, but we also want them to be really fast. So an example might be for using k-means, for example, we would want to maybe try different versions of k or run the algorithm with different choices of k, maybe k equals 10, k equals 15, k equals 20. Here we may be able to do this inside of a lower dimensional space. So going from 20,000 features to like the top 500 PCs or something. Here, these matrices may or may not be able to fit into memory, depending on like if you have 10 billion cells or if you have like 2 million cells. But those are the goals. Okay, so I've talked about k-means. Hopefully everybody here knows what k-means is, but for those that don't, k-means formally is defined as taking a set of n observations. So let's say we have 1 million cells, and I call that matrix X. With a certain set of features, maybe 20,000 features, and a user-defined parameter k. So k defines the number of groups that we're going to partition our number of observations into. So formally, k-means tries to minimize this within cluster sum of squares criterion, where again, X is our data matrix, mu I is the centroid profile for kind of like the I group or I cluster, and we're after trying to estimate those centroids. So in practice, like the way that typically most people estimate this in standard programming languages is based on something called Lloyd's algorithm. It's an iterative algorithm based on 2 steps, where we first say either with some kind of clever way of doing this or not, maybe it's just random assignment. We just say like for a given set of centroids, assign a random set of centroids maybe. Assign each observation to its closest centroid. And then now that you have those labels for each observation, let's just then recompute those new centroids based on all the observations in that have been assigned to that centroid. We just iterate back and forth until convergence. This is the standard way that k-means works. So some challenges with k-means. It's really great. I mean, I'm not trying to say anything negative about it. The downside is, one, it requires all the data to be read into memory. So that's a heavy lift if you got 10 million cells. Second, you have to store pairwise distances between each observation and its closest centroid, right? So again, those pairwise distances are going to become really big when you have lots of cells. Does that make sense? So again, the algorithm works great, just not very scalable. So Scott Scully from 2010 published an algorithm called mini batch k-means. It's a really cool, simplistic idea that sort of makes k-means very scalable, for example. So instead of the algorithm iterating between the assignment step and the 2 steps, the idea is you take, for example, a random subset of the data. They call this a mini batch. So let's say it's 1% of the data, or maybe 5% of the data. Or it could be an absolute number. It could be 100 observations, it could be 500 observations. It's user defined. So I'm throwing another parameter at you. So now you have to pick k. You also have to pick this mini batch size. But so let's say you pick 1%. So at any given point in the algorithm, what the algorithm does is it says I'm going to load in 1% of the data into memory. Or if it's already in memory, I'm just going to use 1% of the data to calculate my pairwise distances. I'm going to estimate my centroids, and I'm going to throw that out. And then I'm going to load in the next 1%, update my centroids, throw that out. Load in the next 1%, update my centroids, and throw that out. So over the course of the algorithm, you do traverse over the whole data set. And at the very end, once you have your final centroids, there's a final assignment step that allows you to assign each observation to its closest centroid. So what does this get you? Well, in practice, there's no need to store the whole data set in memory. It works very easily with on-disk data formats or data formats that are in memory. At each iteration, you only need to store the distances between the observations in the mini batch in those case centroids, which is much smaller. And then also it makes it a really natural candidate for on-disk data. So underneath the hood, mini batch k-means, for those that are interested, it's doing stochastic gradient descent. And I'm happy to go into more details if people are interested. So existing implementations. So I thought this was a really great algorithm, and I wanted to be able to use it in practice. There are some existing implementations at the time. One was in the cluster R CRAN package. If you go to use it, the input says it must be an base numeric matrix type, meaning that you have to have the data stored into memory. So that wasn't great. So if you wanted to use these nice like file formats, it's not possible. There is a version of Python that you can use, for example, in scikit-learn called mini batch k-means, and the input data can be stored in HDF5. However, if you go to actually use the algorithm, there's a step at the very beginning that just feeds in the whole matrix right into memory. So we hit some issues right away. But it does do like the clever, the point of mini batch, you know, to like, you just use 1% of the data. You say like, I only want to use 1% of the data to calculate those pairwise. It does do that, but it does get the whole data set into memory right off the bat. So we implemented our own version and put it into Bioconductor. It's called MBK-means. Does exactly what you want. Let's say you have a file format, an HDF5 file format with 10 billion observations on disk. Those observations stay on disk, and you decide how much data are loaded into memory. And it will very quickly estimate centroids. So let me just give you a quick taste of that. So we took a data set with 1.2 or 3 million mouse neurons. So these are single cell data sets, and we downsampled. So we started with like 1.2 million. We made data sets of sizes 75,000 to 1 million. So these are different sizes of data sets, just downsampled from that one data set. And then we applied k-means in red, and then mini batch k-means saying, let's store the data in memory. Like maybe somebody doesn't want to deal with an HDF5 file. They're like, no, I have enough memory to store my data into memory. Or if you were like, no, okay, I'm really trying to HDF5 file. So this is the version of mini batch k-means with data stored on disk, data stored in memory, vanilla void algorithm k-means. So we tracked the max memory used over the course of the algorithm and the elapsed amount of time it took to run those algorithms. As you can see, k-means is like, you know, a memory hog, as you might expect. Maybe actually means even if you like don't want to deal with HDF5 files, like it does a pretty good job of saving memory usage and speed. If you just simply replace like k-means with mini batch k-means. And then if you are willing to like use HDF5 files, or if that's what you're given, like you can cluster 1 million cells in less than 2 gigabytes of memory in under 10 minutes. And like this is going to stay flat because we're thresholding the amount of data that we read into memory at any given point in time. So like you could have 2 million cells, 5 million cells. Like if we just keep the percentage of this batch size small, it will stay flat. So trade-offs, what are the catches? So I talked about this user-defined parameter, the batch size. So whenever we talk about speeding up algorithms, usually there's a loss of an accuracy. So the take-home message is that mini batch k-means is as accurate as k-means for a large enough batch size. So here I'm showing you absolute batch size on the x-axis. So for data sets of size 5,000, 10,000, and 25,000, just give you some flavors of different sizes of data sets. So here's like I'm reading in 250 observations into memory. I'm reading in 500 observations into memory at a given point in the algorithm due to the batch sizes. And then this is the within cluster sum of squares criterion, which is often how we use to estimate the accuracy for k-means. And so what you see is k-means is flat. That's good. K-means is not flat. That would be bad. And then you see mini batch k-means and blue and green. They just have noise or estimate for small batch size. So you could technically do mini batch k-means with like one observation at a time. You could read in one observation at a time and you would just have very noisy estimates, right? And then as you load in a larger batch size into memory at any given point in the algorithm, what you see is very similar estimates of within cluster sum of squares to k-means. So again, the take-home message is mini batch k-means is as accurate as k-means for larger batch size. In our experience, 500 is about right. And one question I often get is like, what happens if you have rare cell types? If anybody wants to talk about that at the end, I'm happy to answer that question. Basically, our recommendation is just to pick a larger batch size if you have really, really rare cell types. So happy to talk about that if you want. Okay, so we did apply this to our full 1.3 million cells. This is a UMAP representation of the data shown in hexbins. So I find hexbins to be a particularly useful way to visualize lots of observations. So the idea is you just like grid up the U space into a little hexbin, and then you can color based on, say, like the majority of points in that hexbin. What often happens when you have these maps is there is a lot of overplotting that happens, and so this is a really effective way to visualize our data. This is built by the StephanieHicks package for anybody who's interested. And so the colors represent the cluster for mini batch k-means. So how do we calculate that? We took those 1.3 million cells, we applied the top 50 principal components, and then we applied mini batch k-means on top of those 50 principal components. And we did this 16 different times to try out different versions of K. That whole process took 3 minutes. The state of the art in both Python with ScanPy, which is down here, and then the bioconnector version, which is up here, took 35 minutes and 48 minutes respectively. Also use a lot less memory. Okay, so that's kind of the end of the mini batch k-means portion. There's a paper on class computational biology, and I'm happy to talk about it. If anybody has any questions, I'm happy to continue on. Okay, I will continue on. Okay.
Two runs of the block k-means weren't exactly identical as much as their answer.
Yes, correct. Yeah, so there is a level of stochasticity and randomness that you introduce by like choosing random subsets of the data when you read them into memory. Okay, so just to like briefly highlight some other flavors of applications that I work in the world of single cell, and then I'll transition into spatial next. One, I've collaborated with a graduate student, now a graduate of the CS department at Hopkins, Daniel Baker and Ben Langmead, to develop a fast k-means ++ center finding algorithm. So this is called Minicore. There's a Python library available on GitHub. I don't want to go into the details. If you want to know the details, I'm happy to talk about it afterwards. So the idea is we borrow the idea of mini batch k-means to be able, or mini batch in general, to be able to find better centroids for k-means. And so here I'm showing you the in red and green, 2 different versions of Minicore compared to the scikit version of k-means ++, finding those centers. And you can see that for different sizes of K, we are faster. I've worked into other areas called dimensionality reduction. So I mentioned principal components analysis. This is a classic area or classic method that people use in single cell. Turns out it's not so great. So for those that don't know, principal components analysis was designed for Gaussian data, and single cell data is not Gaussian. It's count-based. It has a lot of zeros. As I mentioned, that matrix at the very beginning has anywhere between 50% and 99% zeros. So let's say you took a set of cells that you consider replicates. you expect not a lot of biological variation between them. And then you do the standard normalization and log transformation. This is what happens when you apply PCA. So PCA, the first PC, will almost always try to discover the fraction of zeros in your data. And this is noise, like this is uninteresting noise. So like somebody may say, "Oh, I found a novel cell type here and a novel cell type here." The reality is you just found the cells that have more zeros than less zeros. And again, this is a fact because PCA was not designed for Gaussian data. It doesn't know how to handle those zeros. So we designed, or we extended PCA to what's called GLM PCA. So the idea is instead of picking the Gaussian model, we pick a Poisson model or a negative binomial model or a binomial model, and we just extend PCA for counts data. If you want to know more about it, there's a genome biology paper that was led by Will Towns. And the idea is do not see this like weird artifact of the first PC or the second PC finding the fraction of zeros. And this package is available on CRAN and in Bioconnector. The last thing I'll say in this area is I'm really passionate about reproducible research. So I've developed machine learning standards in the life sciences, or reproducibility standards for machine learning in the life sciences. This was in collaboration with Casey Green, Florian Markowitz, Ben Lee from UW, and many others. I've written reviews about reproducible research in public health. So I'm in a school of public health, and I'm really passionate about this. And one of our kind of like our flagship journals, we have this thing called Associate Editors for Reproducibility, and I joined it a year or two ago, and it's been a lot of fun to help make the statistics community more open in terms of being willing to share their code and make their analyses more reproducible. Okay, so that's kind of the end of the single cell section. Know where I'm at in terms of time. Okay. I was going to shift into some spatially resolved transcriptomics, unless if there are any questions that people want to talk about, I'm happy to talk about that. Okay, who's heard of spatially resolved transcriptomics? Some of you? Okay. All right, so this came online commercially, I would say at the very end of 2019. So we're kind of still in the very new area or the new world of exploring what is spatially resolved transcriptomics and like what are methods that need to be developed versus don't need to be developed. But I will say these methods or these experiments to generate what I will describe as spatially resolved transcriptomics, they're really hitting their stride. So in terms of things you might have heard of, there's two big categories. One are image-based and one are barcode-based, where that's followed up by like standard next generation. Some examples are something like MERFISH, SMFISH. These are all examples of image-based spatially resolved transcriptomics. Or you might have heard of SlideSeq or 10x Genomics Visium. These are examples of barcode-based technologies. And it's really quite exciting. So in 2020, this technology was labeled the method of the year by Nature Methods. And there are a lot of open questions for how to analyze these data. So I'll describe it for you like this. Imagine we have like a postmortem human brain. Apologize in advance for everybody who just ate lunch. Imagine doing like a deli slice inside of that brain and then like just laying it flat in a 2D space. And then it's either imaged or the RNA is bound to spatial barcodes in an XY plane and then washed off and then sequenced. And then you can like map back where each RNA was in this XY plane. Those are the two main types of technologies broadly. But there are a lot of now questions. So like, let's say, how do you store this data? So now we have maybe images and counts. It's not just a standard count matrix. How do we do clustering in an XY plane? Do we just apply mini batch k-means and ignore the XY plane? Because two observations in an XY plane that are sitting next to each other, you could likely use that information to help you figure out what the domains are, for example. Or so do you just like take single cell methods and throw it at it and like hope that's good? Or do you need to develop new methods? So this is a really exciting area. If you're looking for thesis topics to get into, I'd be happy to suggest lots of ideas. Oh yeah, I had a schematic, right. So So spatial-resolved transcriptomics, like you can think about it as we go through like the history of time for how we have been able to, capture this RNA. It's basically like being able to put the Legos together in a 2D plane and be able to now figure out kind of the spatial domains for where cell types map to. Does that make sense? Really like Legos, you can't tell. Okay, so the first thing that my lab worked on was infrastructure. How do we store these data? I know it's like maybe not the most exciting thing, but for us, it's really critical. So at the very beginning, when this data was released commercially, everybody has their own flavor for like how they're going to store these data. Within the world of bioconductor, I thought it was really important to sort of like have people gather around one particular format. So we proposed what's called the spatial experiment format. It's familiar to you if you've heard of the single cell experiment class. That's shown in gray here and right here. That is essentially this spatial experiment class is an extension of the single cell experiment class, whereby in addition to like the counts that you might have in an XY plane for a set of features, row data, call data, reduced dimensions, you also have a space to like place the XY coordinates for each of the observations, and then a space to store all the images that maybe your experimenter, your collaborators generated. So now we have this really nice class to be able to work and generate new methods to analyze these data. So I was first introduced to spatial transcriptomics in a collaboration between researchers at the Lieber Institute, so Kristen, Leo, Harry, and Andrew, who's now left for industry in Boston, which I dearly miss, along with genome-wide and co-senior postdoc in my lab with the summer. So the idea was we wanted to take 3 postmortem membranes. These come from 3 different donors. We looked in the anterior region of the brain. So this is what they call a slab. Lay that slab flat. Now zoom into just this tiny little square here. And this is the only thing that actually gets sequenced. And in this particular area, it's a very well studied area in the human brain called the dorsolateral prefrontal cortex. What? DLPSC. We know that it has 6 very well-known morphological layers and then white matter. So you can see the white matter here. And these are the 6 layers that we wanted to start with. The reason we picked this is because there is so much that's known about this particular area of the brain that if you saw something funnier that we weren't expecting, that would be like concerning. It was mostly just a confirmatory paper to explore this new type of technology in an area that we already have a lot of information about. So the idea was for 3 different donors, we took a tissue block, and then, and I apologize for the big bunch, 2 belly sizes. These are like the 2 spatial replicates here, so belly size 1, belly size 2, and then they went 300 microns into the tissue block, and another 2 belly sizes or spatial replicates that I like to formally call it. Is it clear the design? Okay. So just a little bit more about the technology. So this is what a Visium slide looks like. It's one of the examples of the barcode technologies. You have 4 capture areas. You can have 4 tissue sections on one slide. You zoom into the capture area. You get something pretty cool. In red, you have what's called a fiducial frame. This helps you align an image with the barcode data here in what's called the loop browser. And so there's like a whole alignment step that you zoom into one of these spots. There are 5,000 spots in the fiducial frame, or I guess like several spots in this fiducial frame. You just get these spatial coordinates, and at every spatial coordinate, you have a set of spatial barcodes. That's what's shown here, and then you have a UMI barcode for each unique RNA. That makes sense. And then once everything is bound to the plate, everything is just washed off. Okay, so here's an example of what you get. So this is a piece of the dorsolateral prefrontal cortex in the block itself. This is the white matter. These are layers 1 through 6. This is the tissue on the capture area. In general, here are some statistics about the data that we found. And then this is like what you typically see whenever you start working with spatial transcriptomic data. So in this particular area, this is the gene that's important for the white matter. This is a gene that's important for the gray matter. This is a particular important gene for what's called layer 5. It's associated with synaptic plasticity if anybody's interested, but it's like very clearly lights up in what's called layer 5. And then the reason we know that is because somebody kindly manually annotated each individual spot for all 12 sections. And so this is what layer 5 looks like here, shown in the manual annotations for this tissue section, along with the log gene expression for PCP4. Just trying to give you like a flavor, sort of what it looks like. So this is a zoom out now. This same gene, PCP4, for the 3 different donors across replicate 1, replicate 2, moving in 300 microns of the tissue block, replicate 3, replicate 4. So you get to see a couple of things. Huge donor variation. And then even if you just did like a tissue slice right next to the same tissue block, you have things like this that happen, right? So like, let's say I wanted to take these tissues and align them in these space together, you have these things to deal with. Like, it's not always immediately obvious how to deal with this. You also have wrinkles, so these are like 5.5 millimeters by 5.5 millimeters, and these things are usually wet. So they wrinkle, and you have folds of tissue. So it's like a whole set of computational challenges that I think people who are focused in imaging would be really well defined or really well prepared to help address. So one of the challenges that I'm interested in is how can we computationally identify these genes? So this gene, I've cherry picked for you. I'm trying to make a case that this is an important gene, or that's a spatially variable gene. But I want to computationally identify the genes that are varying in a non-random way in 2D space. So that's what spatially variable gene detection is. So why is this important? Well, these are the features that we are then going to use in downstream analyses. For example, dimensionality reduction, clustering, any kind of downstream analysis. We want to know what are the most important things to be able to do downstream analyses. So this is really like a pre-processing step that is very important for downstream analyses. If you're familiar with the world of single cell, one feature selection method is called highly variable genes, and the idea is to fit the mean variance relationship for each individual gene across all the cells, and you try to figure out the genes that are distant from this blue line, for example. So if you don't know what I'm talking about, this is just a standard way that people do feature selection in single cell. We care about this because it reduces the dimensions of the data from like 20,000 to maybe 1,000 genes. It reduces noise in the data. It improves computational speed. And then sometimes people are just interested in looking at the markers themselves. Okay, so here I'm showing you 6 spatially variable genes. These are the ones I already showed you. They're associated with the layers. These are actually associated with immune and blood. So this one, you can kind of see like a blood vessel. So there are different types of spatial variation. That's my point that I want to get at. So in some cases, you know, you have these like spatial variations that are like big, and some that are small, and some that are kind of like more sporadic. And so we were interested in trying to figure out, do standard methods that exist for identifying spatially variable genes, do they work well? Do they not work well? Are they fast? Are they slow? And so forth. So you could just use like highly variable genes. You could just ignore the XY data, or you could borrow ideas from spatial statistics. So this is like a classic method that people use in spatial statistics called Moran's I, if you're familiar. It's like autocorrelation at a very basic level. There are also new methods. So one that was released in 2018, believe it or not, before like any commercialization of this, by Valentin Svensson, called Spatial DE, and then more recently by Zhao Zhang at the University of Michigan. So the first one is based on classic Gaussian process regression. The problem is this one is particularly slow. And then there are 2 newer ones that are based on kind of like updated models. So instead of assuming a Gaussian model, this set of authors, they assume a Poisson model. an over-dispersed cross-site model or a hybrid model, and this is an entirely different approach. So I'll point out a couple of things. One of the big issues is scalability. So these approaches scale cubically in time as the number of spots. So this paper was designed for data with 200 spots. Every data set that I work with has standardly 5,000. And I just want to point out that 10x is planning to release a new technology with 1 million spots per tissue section. So we'll be doing subcellular resolution by the end of this year. So this is totally not feasible to run. You have to wait 30 minutes per gene, essentially, to figure out if that gene is informative. These ones, or this one is scaled linearly as a function of the spot. So the first two are just horribly slow. And then I will also say we found issues with a lot of these in terms of the genes that they return don't make a lot of sense. So we decided to look at the field of spatial statistics and ask, how can we make this faster? So there's an approach called nearest neighbors Gaussian processes that allows you to basically make Gaussian processes much faster. So there are a couple of ways to make Gaussian processes faster. One is to assume kind of a low rank model. Basically, you just ignore a bunch of the spatial coordinates and you just approximate the distributions that you're trying to estimate on a smaller number, like a random subset of those spatial locations. Or you can look at what's called nearest neighbor Gaussian processes. So this was introduced in 2016. Again, it was designed for maths, we're going to model mortality. It was not designed for spatial transcriptomics. But the idea is you just use an approximate likelihood with a smaller set of nearest neighbors that does approximate the full data likelihood pretty well. It gives a sparse precision matrix. It scales linearly in the number of spatial locations, again, as opposed to cubic for the regular Gaussian processes. And there's some existing R implementations. So this is what we developed in SVG. Because I'm running late, unless somebody desperately wants to see this, is it okay if I skip past some of the details? Yeah, okay. So we developed a method called NNSVG to be able to identify scalable spatially variable genes, and it can scale, to data sets of 20,000 genes with 5,000 spots very easily. There's a preprint on bioRxiv if anybody wants to learn more about that. So this is an example of these spatially variable genes that I talked about. I will say the one thing I'll say is that one of the key insights we found was that when you're fitting these models, you need a gene-specific parameter. So all the methods prior to this, they assume this bandwidth parameter to be common across all the genes. But I showed you that in some situations, genes have a larger bandwidth and some of them have a smaller bandwidth. So we included this bandwidth specific parameter L that's fit per gene. And in this way, we found that if I'm showing you the distribution of L now, estimated across all the genes, MOBP, PCP4 and SNAP25, they all have a large bandwidth parameter, and then HBV and these three, they all have a small bandwidth parameter. So we think that this really helps us in terms of better being able to accurately estimate which genes are most spatially variable versus fixing this at a particular value, which is what other methods do. We're able to accurately recover interesting spatially variable genes. There's a bioconductor package that exists for this if you're interested in using it. Any questions on that? And then one last slide and then I'm done. Okay, you guys are very patient. Hopefully it's interesting. So the last thing I'll mention is this is a new work out of my group this week called Loopy Browser. So one of the challenges we face as we are starting to generate these data is that all of these interesting data come with really big images, and we want to easily share those images with others. So there are a couple of really great tools like ImageJ, Fiji, Napari that are all graphical user interfaces. They're all GUI based. So if I wanted to share my image with somebody, they had to install a graphical user interface to be able to interact and annotate and visualize that image. I wanted to be able to just send somebody a URL. And so what we did was we developed this thing called Loopy Browser, which is a tool for you to be able to interactively visualize, annotate, select, move, and it's all web-based. It's based on a couple of things. So it's based on the Open Layers platform, which is a tool for you to be able to embed apps into web pages. And then also each one of these images, which are TIFF files typically, get converted into GeoTIFF files that are used, for example, in Google Maps, and they're chunked so that when somebody lands on the page, you get one chunk of the data that's downloaded, and as soon as you zoom in or zoom out, you get more data that's downloaded. So the data itself can be local, or it could be uploaded to an AWS S3 bucket. And in this way, similar to the way that you interact with Google Maps, you interact with really large images in the browser. And this is what makes it so fast. So now I want to just show it to you. I'm probably going to break everything. Let's see. You guys want to see Visium or Merfish? Okay. I showed you enough Visium already. This is the browser. And so you have the histology image and the background, and these are the manually annotated regions, and you can control the color contrast between them. You can turn off the annotations. You can turn them back on. You can also, oh gosh, I'm definitely not able to see anything. You can search for your favorite gene, like PCP4 that I've shown you quite a bit. And now you're seeing, hold on, did I type that right? Maybe, okay, MOVP. Sorry, it's very hard for me to see that, you guys. MOVP, so you can see all the white matter is down here, for example. You can annotate different types of things, like if let's say you wanted to try and circle. This is going to work much better on my laptop. Okay, if you guys want to talk about it later, we can, but it's the Loopy browser. It's really cool. I would love to talk about it if people are interested. Yeah.
I have a question in the previous section, but this is a good time to ask it. You touched on this. How well are you able to do the biological replicas when it comes to images, aligning them? And,
In sequence, if I take two tissue sections and I want to line them.
that's right. And then using the manual notation and going back to the gene expression, trying to readjust the manual notation based on the gene expression.
Yeah, we don't do very well at all. And so it's pretty hacky, I would say, in terms of being able to line them in Z-space. So there are two methods, one called paste, out of faculty member at Yale, getting his name. And then there's another one from Barbara Engelhardt. This is at Stanford, and it's Gaussian process based, and it's horribly slow. So there are approaches, but again, they don't work very well, and they're not what I would call spatially smooth. So if you try to align them in Z-space, you get really rough domains and really rough output for what you would expect. Does that answer your question? Yeah. So again, I'm trying to sell spatial transcriptomics to you. I think there's a lot of room for methods development if you're interested. Okay, and that's it. So thank you, everyone.
We have time for questions. Questions for Stephanie?
I am novice with the computer stuff, but I know cells and I know the brain. So I look at your example and know that within layer 5, there are a lot of different cell types, and they're intimately interacting with one another, and they're very close to each other in space, right? So even if you broke down, okay, well, there's neurons, there's glia, there's blood, there are 10 different types of neurons, just in layer 5, for instance, and different types of glia. So, and then that's, okay, the single cell data where you lose the spatial resolution might pick up that better. So, what is the interface, between the two, or is this one of the problems that you're talking about? So how is there a way to say, I know that there are these cell types and some of them can be defined by, this glial gene or this neuronal gene, and then incorporate that into the spatial analysis so that you get that multi-layer.
Yeah, so the term that people in the community have rallied around is what's called spatial registration. So the idea is you take a single cell data set and you can consider like this a map, so to speak. Like, let's consider this a map. And like I want you to like know, for example, if I've got a glial cell, where does it map to the best in an XY coordinate plane? Or if I've got an excitatory neuron, where does it map to the best? And so there are a lot of great single cell data sets that don't have any kind of spatial data. There's a lot of phenotypic information about them, either at the cell type level, or maybe they come from neurotypical donors versus schizophrenic or bipolar MDD, for example, and you want to figure out where do they map to? Like, that's the term that people are using, and there are methods that are starting to be developed for that. So that's just like purely taking a single cell data, quantitatively, algorithmically figuring out where it maps to and how you talk about approaches. The other approach that people are taking is deconvolution. They're borrowing the idea from bulk, and they treat each observation here, each spot, which is like a mixture of cells. So it can be anywhere between like 0, which is typically like neuropil in the brain, for example, to maybe 10 cells per spot. And it's a mixture. So then the question is, if I have single cell profiles, similar to if I have like single cell data that I want to deconvolute from a bulk tissue sample, can I deconvolute each observation and imagine like having a little pie chart at each one of these spots? And that's actually what people show. And you can see like, inhibitory neurons, excitatory neurons, white matter, you can see it like recovered, that type of information. But it's mostly proportions. Tangram is one that will actually like give you counts, but in our hands it's really. So I can talk about this, but there's like a whole variety of approaches that people are trying to use to integrate single cell with spatial. But it's pretty heuristic and like rudimentary early days.
Okay, so thinking about the highly variable genes in like a spot-based method like this versus in the imaging-based approaches where you can get like cellular, are you guys working on, or are you aware of anyone who's doing like subcellular?
Yeah.
variability?
So MERFISH is the classic approach that people point to for this. The problem is it's so challenging to implement in your own lab. Like you basically have to go to Boston and do a postdoc to like be able to like implement MERFISH in a lab outside of that, or you have to have like very specialized person to help you run that. So there are new technologies that have just come out commercially available. So 10x's version of this is called Xenium, where you can capture 400 genes. And you can do now spatially variable gene detection at a subcellular resolution. There's nothing stopping you from doing that. But again, this gets you the full 20,000 genes that and MERFISH only give you like 400. So the idea is, if you were like setting up an experiment, like if you want to discover, you probably want to do something like this, where again, at top level, it's not single cell level, but it would allow you to figure out what are the genes that seem to be most important, and then you do them with like a specialized subset of genes, for example, and then or MERFISH, if you can figure it out. And then like you could, for example, identify something that's going on cellular. That's I think how like the community is recommending doing this. Like you first discover with like not perfect data, not perfect in the sense that it's not truly subcellular. That's what everybody's beef is with Visium. It's like, but it's not subcellular. Like, okay, well, better than not. Yeah. Other questions? Yes.
So you referred to the problem of Z registration, and what are the approaches that exist for trying to do that? I mean, do they try to align the images? Do they try to align the gene expression? Do they try to align both simultaneously?
Yeah. So the one I mentioned by Barbara Engelhardt, they align the gene expression. So they take the spatial coordinates and they try to work the gene expression and then just align the spots using Gaussian processes. And so it's really slow. Then I can't remember the person's name for me. Called Paste, and he just released a new algorithm called Paste 2. Somebody please tell me his name. It's gonna bother me. Anyways, that approach I am less familiar with, But I know it works much faster because the person that I'm working with who's implementing it seems to tell me that it's working much faster, but I don't know what it, like under the hood, how it works, unfortunately. I do know that it doesn't really give you like spatially smooth expressions. So let's say you align them, it kind of like treats them still independently. And so depending on what you want to do with that alignment, like I could imagine issues arising.
I see. So could you like try to use image registration techniques to constrain the space?
Yes, I just don't have enough time in my day. Like, it would be amazing if we could get into this field. Yes.
So Cecilia Lowe, who's now at Pittsburgh, she used to do this many years ago now, about 15 years ago. So she had some software that worked on the image. She worked with hearts, which are easier to register than brains, but you might want to look her up, and I don't know if she's still active.
Thank you.
but-
Thank you.
All right. I hate to interrupt, but we have another group that needs to come in. Thank you so much. If anyone would like to continue